Friends, welcome you all on the behalf of Rahul Anand Biology Tutorials. So, as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand Biology Tutorials. So now, we are going to study about the very interesting topics which are often being asked in terms of your NEET, often the aims, often the All India Pre-Medical Examination. So these are what? So these are particularly belonging from the molecular basis of inheritance chapter. So today we are going to study about what? So today we are going to study about the central dogma. So when we are talking about these cases, means what are actually the central dogma? Remember, so it tells us that in this case the genetic material flow will be occurring in terms of what? So in this case the genetic material flow will be occurring in terms of the one direction only, or either it is unidirectional. In this case what will be happening? So in this case the genetic information will be literally flowing from what cases? So it will be literally flowing from the master copy DNA as well as towards what? Towards the working copy RNA in terms of what? In terms of the biological system. As well as when we are talking about these cases, means what are actually the DNA. So you are very clear about that. Remember what are actually the DNA? So DNA literally means the deoxyribose nucleic acid primarily. When we are talking about these things, so your DNA are literally being made up of the two of the polypeptide chain. As well as when we are talking about these cases, means what are actually these things. Remember, so these are the ribbon thread like structure, as well as these are particularly being attached in terms of the antiparallel direction. Often in this case we will be having what things? So often in this case we will be having the in terms of the helical structure with reference to how much degree of the angstrom. So these are the 34 angstrom of course, as well as in these cases we will be having the nitrogenous bases such as what? So we are having the in terms of the adenine, then we are having the thymine, then the guanine, as well as the cytosine. As well as when we are talking means how does these in terms of the adenine are literally being attached with reference to your thymine. So it is being attached with reference to the two of the hydrogen bonds. As well as when we are talking about the second case, means how does these guanine are literally being attached with reference to the cytosine. So it is being attached by the help of the three of the hydrogen bonds. Now, so talking about these things, so DNA are of course regarded as what? So these are literally known as the genetic material. So you are often very clear about that. Means what are actually the genetic material. Remember, of course. Now, so when we are talking about the another cases, means in terms of these things, in terms of the DNA. So there was the particularly scientist played an influential role in order to do what? In order to propose the 3D model of the DNA. So he was actually what? So he was nothing else but the Watson and Crick. Often you are very clear about that. We are often understanding about these in terms of the Watson as well as the Crick. So very clearly understanding. So he played an important role to propose what? To propose the 3D model of deoxyribose nucleic acid structure, of course. Now, so when we are talking about these cases, means in terms of the Crick. So here we are often going to study about the Crick. So his full name is actually what? So your Crick full name is actually the Watson Crick. Now, so he was actually the biochemist. Remember, so he particularly studied the biology. Often he particularly studied the chemistry. Now, so when we are talking about these things in terms of the another cases, so understood means what are actually the deoxyribose nucleic acid. As well as when we are talking about these things, means how these particularly things are literally being done, mean how these processes are literally being transformed in which the master copying DNA are literally being copied with reference to what? With reference are being transferred in terms of the, tell me, in terms of the, in terms of the working of the RNA, in terms of the working copy RNA means the ribose nucleic acid. But when we are talking about these things, means what are actually the ribose nucleic acid. So you are very clear about that. Means what are actually these? So these are the single strand, of course, ribbon-like structure. But in this case, the pairing substances or either the ribbon thread-like structure will literally not present. So these are known as the RNA, means the ribose nucleic acid. Often when we are talking about these things, means in terms of the ribose nucleic acid. So your RNA is not the genetic material. Remember what are actually the genetic material. But here we are having the critics or either the exception. Why? Because biology is the science of exception. Now, so when we are talking about these things, means what are actually the genetic material of your meant of the ribose nucleic acid. So these are what? So these are the reoviruses, often popularly known as the retroviruses. But when we are talking about these things, means what are actually the retroviruses as well as the reoviruses. So these are popularly the viruses which are literally being present in terms of the AIDS diseases. As well as you are very clear about that, means what are actually the full form of your AIDS, means the acquired immunodeficiency syndrome or either the viruses. So these are particularly having the spikes like structure, of course, in terms of what? In terms of the retroviruses, as well as these are often having the inner structure in which it will be having the P18 then the P24. Talking about the case of the P24, so these are literally being present in terms of the outer cell, as well as the P18 will be present in terms of the internal cell. Often when we are talking about these things in terms of the retroviruses, so these are having the spikes like structure. Okay, fine. So these are playing important role to recognize the helper T cell. After that it will be doing what? So after that it will literally trap the helper T cell or either damage the helper T cell. Remember, as well as weakening, playing important role to weak down the immune level or either the tuned amount of the healthy status in terms of the body. So these are particularly being transported by the help of the certain of the transmission, such as the sexual transmission. Okay, as well as the another symptoms such as usage of the infected needles or either being present with the in terms of the infected person, rather maintaining the sexual contact with reference to the infected person. Now, so talking about these things means what? Means how this process of the dogma process are literally being employed. So you are having these things or rather the three very important process which are literally being employed. So these are fully known as what? Tell me. So these are the here meant of the replication, then the transcription, often the translation. Talking about these things means what are actually the tell me means what are actually the here meant of the replication. Okay, fine. So in this case the deoxyribose nucleic acid will be copying in terms of the deoxyribose nucleic acid. On the second hand we are having the transcription. So when we are talking about the case of the transcription, so in this case what will be happening? So in this case the here meant of the deoxyribose nucleic acid will be copied in terms of the here meant of the mRNA. Remember, just we have understood about the ribose nucleic acid, but when we are talking about the mRNA, so these are what? So these are the messenger RNA. In the third, this will be particularly being copied with reference to what? So this will be literally copied with reference in terms of the polynucleotide or the protein. So these are known as what? So these are fully known as the translation process. Remember, when we are talking about the case of the protein, so protein are what? So protein are the macro. Macro means what? It will be the larger understanding. So it will be the larger quantity of the amino acid. Okay. But when we are talking about what are actually the amino acid, just reverse the process. So amino acid will be what? So amino acid are the macromolecules of, tell me, amino acid are the macromolecules of your protein. Now, on the other hand, we are having here made of the polypeptide. Find here. So polypeptide means what? So your polypeptide means when many of the nucleotide combine, it will result in the formation of the polypeptide. Okay. Fine. Now when we are talking about the another cases, such as what? Such as means how does these process are literally being employed. So you could often popularly find here the structures of the DNA, then the RNA. Often here made of how are these literally proteins or either the polypeptide present. Find here. So here is the DNA, then the mRNA, then the protein. Now, so when we are talking about these things means what are actually the central dogma. So these central dogma hypothesis was literally being proposed by whom? So these central dogma hypothesis was literally being produced by the Francis Crick in terms of what of the era. So it was actually the, tell me, so it was actually the here mid of the 1958. So he played an important role in order to propose these hypotheses in terms of the central dogma, understood as well as very clearly understanding about that. Find here. So here is the 1958. So just as we have understood about these things means how these processes are literally being employed or either carrying out or either taking place. So in this case we will be having what? So in this case of course we will be having the here primarily main term the DNA means the deoxyribose nucleic acid, then the RNA of course, as well as we will be often having the protein or either the polypeptide of course. But when we are talking about these things means when the copying will be just done in terms of what? In terms of the DNA to DNA, it will be known as what? So it will be popularly known as the here main term the replication. Often very clearly understanding means replicate actually means what? So it means that something is being copied into the same amount. So when we are talking about these things, so these are often understood as in this case what will be happening. So in this case one nucleotide or either the one nucleic acid will be copied in terms of what? So in this case of the replication, one nucleic acid will be copied in terms of the another nucleic acid in all the three or either particularly the two of course in terms of the what? In terms of the replication, then here in terms of the transcription. Why? Because we are having the DNA. In this case, we will be having the deoxyribose than the nucleic acid. Understood? Then again, we are having the RNA. Means what? The ribose than the nucleic acid. Now, so in the copying will be done in terms of the DNA to DNA. So it will be known as the replication. As well as when the copying will be done in terms of what? As well as when the copying will be done from the here in terms of the DNA to mRNA, or either the messenger RNA or either the RNA. So it will be known as what? Tell me. So it will be known as the transcription. Then in the third, when the mRNA transfer will be done with reference in the case of what? In the case of the protein or either the polypeptide. So it will be known as what? So it will be known as the translation process. Now, so when we are talking about these things in terms of the central dogma. So central dogma, as well as when we are talking about these things in terms of the genetic material, of course. So these genetic material are literally being present in terms of what? So these genetic material are literally being present in terms of the central dogma. As in terms of what?